Dear student, myself Dr. Sanjay Kumar Srivastav, and today's my topic is the practical of DLC, differential leukocyte count, how it has to be done in the laboratory. A laboratory specialist puts a drop of blood blood sample on a clear glass slide and smear it to spread the blood around. Then they stain the blood smear with a dye that helps to differentiate the type of white blood cells in the sample. The lab specialists they count the number of each white blood cell through the blood cell counter manual procedure. So, how it could be done? After the preparing of smear, you should air dry and then go for Leishman stain. And there are few stains are used, then that is Leishman stains, Wright stains, Giemsa stains, field stains. The preparing the slide, collect drop of blood on the end side of a glass slide, spread the blood drop with another glass slide by placing it at an angle of 45 degree and move sidewise. Hold the spreader firmly and move it on the previous slide to the other end of straight line with same force and pressure. Allow the glass slide to dry after formation of blood sample and then go for staining. After the staining, you found the types of leukocytes in the smear. It is of 5 types, that is neutrophils, lymphocytes, monocytes, eosinophils and basophils. The procedure is the leukocytes are classified into various groups depending on their size, features of the nucleus and features of the cytoplasm. The WBC exists in two forms, that is granulocytes and agranulocytes. I think the theory part of this subject is taken by Dr. Girish Chaturvedi and he has been told to you also, and the granulocytes are further classified as eosinophil, basophil, neutrophil, while agranulocytes shows lymphocytes and monocytes. Sample required, the best sample is EDTA blood and it may be prepared fresh peripheral blood smear by fresh blood, and you can use another anticoagulant, that is heparin citrated blood also, but good way is only fresh blood or EDTA blood. So, requirement of the materials are cotton, micropipettes, glass slide, distilled water and stains, decolorizer, that is distilled water, and there are 4 common stains used to the staining of slide, that is as I told you, Leishman stain, Wright stain, Giemsa stain, field stain. So, after staining, observation the slide and counting of the cells can be done by the prepared slide is under low power of microscope and choose a good quality slide. Then identify different types of WBC under medium power. Then go draw a table with 10 boxes both on horizontal and vertical axis. On a observation notebook or use hand counter to count the cell. Fix the slide on the platform and choose an area towards the corner. Count the different types of WBC found on the table in an abbreviated. Move downwards and in chain like manner till 100 cells are counted. Here I have shown you the picture of WBC. Here is A basophil, B lymphocyte, C monocyte, D eosinophil, E band cells and F neutrophils. These alphabets are indicates the type of WBC found in the peripheral smear. You can look here it and understood very clearly. And what is the normal reference range in male and female? So, here a chart which I have shown you, you can see here neutrophils are polymorph in male 40 to 75%, in female 30 to 75%, and lymphocytes in male 20 to 50%, in female 20 to 50% also, monocytes 2 to 10% in both, and eosinophil 1 to 6 in both, basophil 0.3 to 1% in both, that is male and female also. So, the monocytes have a longer lifespan than many white blood cells and help to break down bacteria, and lymphocytes they create antibodies to fight against bacteria, virus and other potentially harmful invaders. Neutrophils they kill and digest bacteria and fungi. Eosinophils and basophils are the found in allergic condition, and basophils are found in cancer also, blood cancer also. Here you can see a peripheral smear drawn on a slide and stained here, and this smear one is thin layer and another is thick layer, in such a way allow to various blood cells to be examined microscopically. So, these are the differential leukocyte count. If you have any problem to understand it, then you can ask me question on my YouTube channel.